Hi, my name is Allison, and I'll be going over the protein synthesis process. So the first part of the protein synthesis process is transcription, and transcription is when you convert like a sequence of nucleotides from DNA to mRNA. So in this case, as you can see, there's the RNA polymerase that is basically copying down the nucleotide sequence from the DNA to the RNA transcript. So what is DNA? DNA carries genetic information, and this particular region can be called a gene, which is just a sequence of nucleotides. Here we see the RNA polymerase. Again, it is copying down the nucleotides or the complement of the nucleotides onto an RNA transcript, and it's copying from what we call the template strand. And here we can see in transcription how that works. So here's the template strand of DNA and the mRNA. In the mRNA, we can see a difference. There's uracil in the mRNA, while there's thymine in the DNA. So uracil only appears in RNA, and thymine only appears in DNA. And as you can see, they're just the complements of each other. Next, so once that is transferred onto the RNA transcript, we now have to attach a poly A. So poly A is just responsible for making the RNA molecule more stable and to prevent its degradation. Also, it allows the RNA to be exported from the nucleus and translated into a protein by ribosomes in the cytoplasm, which we'll get to later. And the 5 cap, as you can see here. So the 5 cap is added to the first nucleotide, and it's a modified guanine nucleotides, so it protects the transcript from being broken down. Now, going back to the pre-mRNA, this is actually not ready to be transferred out of the nucleus yet, because there's exons and introns. So the exons go on to be covalently bonded to one another after the introns are removed. So the introns are not expressed in the final messenger RNA sequence. So these are going to be removed, and then these ends will be covalently bonded with each other. So after, we can see that it will transport out of the nucleus. So this part was transcription, the first half. Now we go on, and transcription occurs in the nucleus. Now we go on to translation. So what we have in translation is we have the mRNA that is transferred all the way out here. And basically, the mRNA now goes through these so these ribosomal subunits combine to make this big unit, and the ribosomal subunit, the big one, has 3 different parts. So here's the E site, the P site, and the A site. So the A site is where the molecule comes in. The P site is where the molecule binds, and then the E site is where it exits. So first off, we have amino acyl tRNA synthetase. So what this is responsible for is actually, it basically it's an enzyme that matches up the amino acid with, uses the codons to match it with an amino acid, and then attaches it to a tRNA. And this requires ATP, so it uses up energy. Now you have the tRNA, and now it's ready to go into the ribosome. And what it does is it basically it makes the anticodon. So here we see an anticodon, and here we see a codon. So the codon is part of the mRNA, while the anticodon is attached to the tRNA. And what the anticodon does is it's just the complement of it, like we saw in this transcription part. Now let's move. So what this tRNA does is it basically kind of like it enters through, remember the A site, and then it, in a way, it like kicks off the other one, and then let's see, and then this one is like it goes to the exit site and it moves away after having dropped off its amino acid, and then this one moves in, and then later another tRNA will come in and kick it off as well. I forgot to mention also, with the codons, 3 codons code for 1 amino acid. And here we see, as it drops off the amino acid, the polypeptide grows. So here we see, as like more and more different amino acids are dropped off, that's how we grow the protein. Yeah, and then it just keeps, this is started by a start codon, which is methionine, and it's stopped by a stop codon. Yeah, and as the peptide grows, this is called elongation, and also another one is peptidyl transferase. That's those like little black lines that you see here that basically connect the 2 amino acids together. Yeah, so that is protein synthesis. Cool. I hope you enjoyed. Thank you.